Hi, everyone. Welcome to the Group 6 Fly Lab presentation by Amal Gowda, Min Tran, Divyesh Trivedi, and Maria Grill.
So a quick introduction to mapping. So 3-point mapping, according to a research study done in 2006 on gene mapping in worms, is the process of crossing a mutation into a strain with linked morphological markers that are on the same chromosome to generate a heterozygote. So in order to successfully carry out a 3-point mapping experiment, at first we have to cross an individual that displays the dominant phenotypes out of the phenotypes that we are studying with a known homozygous recessive individual. And that dominant individual for those phenotypes that you're looking at, we don't know if it's a heterozygous dominant or a homozygous dominant just by looking at it, because that's impossible. And the results from that cross can show us which organisms display the wild type and the mutated phenotypes after further examination. So we were able to examine both females and males that were present in the cross analysis. So we actually got to see sex differences between the flies as well as the phenotypic differences between the two. And the markers that we used to analyze the phenotypes, we used a plus sign to indicate the wild type phenotype and no plus sign, just the lowercase letters to indicate the mutant.
All right, so our hypothesis is that the distances between loci do not affect the accuracy of the mapping data. And the reason for us thinking this way is because of the direct relationship that exists between recombination frequency and the distances between loci on a said gene, and specifically this gene we're looking at. And this is because some crossover events have the ability to affect other crossover events.
So as for the experimental design, the cross that was carried out, it actually occurred before we got the fly vials. So the fly vials that we had received had already been the crossed flies that had happened previously. So the individuals that were crossed consisted of a dominant individual for each of the 3 traits that we were looking at. So wing size, body color, and eye color. And it was crossed with a known homozygous recessive individual. And like I mentioned before, the dominant individual, we don't know just by looking at the phenotype if that individual is homozygous dominant or heterozygous dominant. We just know that it is dominant. And in order to successfully carry out the cross and the analysis and the gene mapping that subsequently follows, there must be a sufficient amount of offspring, which you will see later we absolutely had after the etherization occurred, which segues into the actual etherization of the flies. So what we had to carry out was we got the fly vials, like as seen on the right of this photo here, and we had to prep the etherization tube with some ether alcohol by saturating the cotton ball or the cotton interior of the tube with ether alcohol. And we placed the fly vial after pulling the plug from the top upside down into that other etherization tube, tapped down on the top so that the flies fell down through the tube and were killed by the ether alcohol in the tube, held it there for 10 minutes, and then our dead flies were then able to be examined on a note card after that 10 minutes was complete. And quickly here on our table, we have the comparison of the wild type versus the mutant phenotypes. So as for the wing type, we have the wild type, which is just the smooth wings. And then we have the mutant type, which is the vestigial wings. And then we have 2 different body type or body colors that we found. B+ indicates the brown body, and then B indicates the black body. And then we had the 2 different eye colors indicated by the plus sign, which was the red eyes, and then BW, which is the brown eyes.
Okay, so now on this slide, we're looking at 2 pictures of fruit flies that we etherized and analyzed ourselves on the index card and with the microscope. So on the left, we have figure 1A, Drosophila melanogaster exhibiting all dominant alleles. So that basically means smooth wing type, brown body, and red eyes. And in the other figure, figure 1B, we have a fruit fly that, on the other hand, exhibits all recessive alleles. So vestigial wing type, black body, and brown eye color. And so this is just to give you guys just a bit of context and a sense of what the genotypes that we've discussed actually look like in real life. And it's pretty interesting, in my opinion, to see how they translate over to the real world.
So from here, we have data from our test class, and the data is taken from all the groups on our class. So it shows the phenotypes of the different of the Drosophila from the test class. And in this case, their two genotypes are paired together because the highest number of occurrences is the parental genotype, which is having the three wild type alleles, while the other one has three recessive alleles. So, and from that, if you look at the group that has the lowest number of occurrences, which is the one that has a brown body, normal wings, and brown eyes. And from that, we can understand that the vestigial is between the allele that codes for the vestigial wings is between the brown eyes and the black body. And using this, we can look at the gene mapping of all three of these alleles on the chromosome. So this is a genetic map that we made up. So in this figure 1, we have all three alleles on the same chromosome. As we know before, the vestigial gene is in the middle because of the lowest occurrence, because the double crossing over happens with the lowest frequency. So in this case, we calculated 40.5 map units between brown body, between black body and vestigial wings, and this was done by looking at the data above, adding the two values for which there was a single crossover and a double crossover, and dividing by the total number of flies, which is 595. And a similar thing was done with to find the distance between the vestigial allele and the brown eyes allele. In this case, it was 40.17 map units.
All right, so now on to the evaluation of our hypothesis. So essentially, we were able to make our predictions through the use of chi-squared analysis. And as a result of that, we determined that our p-value was less than 0.001. And that shows significant differences, which essentially means that our map values differ significantly from those provided by flybase.com. And on the right, we have an image of the work we did to do the chi-squared.
So our calculated map distances differed significantly from the known and accepted map data, as Mo just alluded to. So given this information, our hypothesis is refuted because clearly the distances between loci had some type of effect on the accuracy of the mapping data in order to induce a significant difference. So this somewhat aligns with our previous expectations. On one hand, we did indeed expect the hypothesis to be refuted. This stems from the well-accepted notion that as the distance between two genes increases, mapping estimates become more inaccurate due to undetectable double crossover events yielding the same allele combinations. But on the other hand, our data supports the occurrence of negative interference, where the number of observed double crossover events is actually higher than expected. So based on the previously mentioned logic, we would expect positive interference to occur, where the number of double crossovers is lower than expected, which is right now most often the form of interference that we believe to occur in eukaryotes. So these results are rather puzzling. However, our data can perhaps be somewhat justified by the fact that interferences do decrease as the genes in question are located farther apart. And the genes that we're looking at right now are not exactly extremely close, especially between black and vestigial, according to Flybase. However, a large percentage of these results can also definitely be attributed to flaws in experimental design. For example, our sample size was relatively small, probably several orders of magnitude off the ideal, which allows for a higher probability for unexpected results to arise. Additionally, error in correctly identifying Drosophila phenotypes, although marginal if at all, may have occurred. So future experiments may achieve more expected results by remedying these errors.